All right, welcome back to Monkeys on Typewriters on YouTube. We've got a pretty special discussion and presentation today on a theme we haven't covered so far. But if you've watched the previous videos on paradigm shifts in cancer and thinking evolutionarily about that, and also some of the presentations and discussions on COVID, you'll find this directly relevant and hopefully a pretty good follow-on topic. So we've got a couple of other participants today. We've got Rachel Thomas and Maddie Stoddard, and maybe we'll just do a really brief introduction and go around a little so that new viewers know who you are, and then we'll move into the presentation. Rachel?
Sure. Hi, I'm glad to be here. Briefly, my background, I have a PhD in mathematics. I worked in the tech industry for several years. I was co-founder of Fast AI, which created the kind of most popular or one of the most popular and longest running deep learning courses. And I'm now a researcher at Answer AI. And along the way, I did a master's in microbiology and immunology.
And you're located in Australia right now,
Yes, and
so
I'm in.
we're working with time zone differences here. Maddie?
Hi, I'm Maddie. I'm Senior Director of Modeling and Simulation at Fractal Therapeutics. So my work is as a drug development consultant, but my research spans COVID-19, applying evolutionary theory to cancer, um, and now we're dabbling in this immunology space. Pleased to join you.
Great to have you here. Okay, I'm going to turn it over to Arijit and Rachel to take us through immunology as a Red Queen's race. In other words, looking at immunology from the standpoint of evolutionary processes and not just the sort of standard paradigm that has had, as we'll see, some challenges, I believe. All yours.
Great, thanks. So the structure of the presentation will be I'll jump in, do a little bit of talking, then Rachel will take over at some point, and then it'll come back to me. So if you see us switching off back and forth, don't be alarmed. That's part of the plan. So we're talking today about immunology as sort of an alternative view of how things work behind the scenes, which is kind of a bit of a bold statement, right? We're looking at a whole discipline. We're saying, gee, maybe the way that people are framing the fundamental premise behind immunology could be a little bit off. Why does this matter? This matters in practical terms because a lot of what gets said during things like pandemics, during things like drug discovery and development relies very much on having the right view of the world. So if your view of the world is slightly screwball to begin with, that has consequences all the way through. Now, full disclosure, none of us here is an immunologist. We're coming at it from adjacent fields, looking at immunology and scratching our heads. And those of us who have entered new fields and worked in new fields, very often when you bring an alternative perspective to a field, it allows you to ask questions that people who are steeped in the field may not necessarily be asking. So none of this is to say that all immunologists are wrong or that everything about immunology is broken. In fact, today we'll emphasize some really fantastic papers in immunology that we believe have the right way of thinking about it. But it's a step back from the field as a whole, from the immunology we all know and love. Just kidding. Or that we learned at least in school, with all those pathway diagrams, and then asking the question: is this really an appropriate view? So this comes back to some of the conversations we've had previously on this channel with Ryan and myself, between Ryan and myself, around paradigm shifts. And in the context of cancer, the paradigm shift question becomes very, very relevant because you really start asking the question differently. If you view cancer through a different lens, then it allows you to ask fundamentally different questions than if you are locked into oncogenic dogma. So similarly with immunology, it begs the question: is the current paradigm, is the prevailing paradigm correct? What are we looking at, and how could it be different? So a lot of this now that I'm going to be talking about relies on an old book called The Structure of Scientific Revolutions, which many of you who are practicing scientists may be familiar with. Maybe you've read the Wikipedia page for it, and if you haven't, I highly encourage you to do that. Kuhn's book, which came out in the early 60s, was a really radical book because it talked about the practice of science. It talked about sort of doing science under business as usual terms versus when science under business as usual framework starts breaking down, and sort of how that change happens when you shift from one paradigm to the next. So we've slung the word paradigm around quite a bit, and so it begs the question: what is a paradigm? Well, a paradigm is a set of facts that allows you to frame a view of the world. So in Kuhn's book, he says men whose research is based on shared paradigms are committed to the same rules and standards for scientific practice. So because you think of this the world in a certain way, every new data point you gather is filtered through your understanding of the world. So if you believe, for example, in the paradigm that amyloid fibrils are responsible for causing Alzheimer's or pushing Alzheimer's disease forward, then every new piece of data you get, every scientific hypothesis you put in place will be based on that. And so you'll then try putting in place treatments that eliminate amyloid fibrils and so on and so forth. And so everything that you do will then rely on that paradigm. And the paradigms are very powerful because often people don't even realize when they are in a paradigm, they don't even realize that they're actually in that paradigm. And we'll get to that in a little bit. But paradigms are often very, very unspoken assumptions just about how the system works. And so, for example, you could think to yourself that, okay, well, electricity is a fluid, right? And what happens when you change the paradigm is that you change the practice of science. So when people thought that electricity was a fluid, what they thought was, okay, well, then, you can gather electricity in a jar, right? And so they actually built these things called Leyden jars. And Leyden jars are essentially these large physical jars that hold charge. And so they're essentially capacitors. If you go one by one, every element that you see in a capacitor is present in a Leyden jar. So on the one hand, what does it matter that electricity is not a fluid? You can still build a Leyden, you can still hold an electric charge. That's correct. But good luck building a computer with the fluid theory of electricity, because exactly how large a room are you going to need to string together billions of Leyden jars? So changing the paradigm allowed people, when they understood that electricity was composed of electrons, the electronic theory of electricity allows you to do a lot more with it than the fluid theory. If you think about it, even things like Kirchhoff's laws, which are current flowing in, flowing out, you can model, you don't necessarily need the electronic theory of electricity to work with Kirchhoff's laws. It still works with electricity being a fluid, but the right paradigm allows you to do things go much further than the wrong paradigm does. Of course, the wrong paradigm in retrospect can seem quite silly. For example, if you ask people, at a certain point in the past, how does light travel? They'd say, well, it requires a matrix, and it's propagated through the ether. We know that this is not true. Light travels through vacuums. Or what about fire? Well, the ancient Greeks used to think that burning releases a substance that we perceive as fire. But if you weigh an object before and after it's burned, it gains weight. It doesn't lose it. How epidemics spread. People used to think it was caused by bad air, resulting from rotting organic matter. So all of these paradigms today sound silly to us because we know this is not how it works. But it's important to understand that these were widely held beliefs. And so the degree of social acceptance of a belief is not a measure of its correctness. You can have as many experts as you want lining up, swearing up and down about a paradigm, and that doesn't necessarily mean the paradigm is right. So, of course, we know that sometimes having the wrong paradigm in place can create disastrous outcomes in a public health context as well. And so these things are not abstract scientific debates, they have practical consequences. So how then do you know that a paradigm is in crisis? Well, one of the key sort of hallmarks of a paradigm being in crisis, and Kuhn in his book offered several of them, but one of them is that important problems remain unsolved. The second hallmark is that other paradigms offer competing explanations. And the third one is that the paradigm just keeps getting more complex and more vague as you gather new data. So, for example, the ancient astronomers believed that the Earth was at the center of the universe, and the sun and the stars and the planets all revolved around the Earth. Right now, when they measured the trajectories of planets, they found that every once in a while, if you're watching Mercury kind of moves backwards and then moves forward again. And that retrograde motion of Mercury had to be accounted for. So what they did is they took the orbit and they put little cycles on them, epicycles. And over time, as they gathered more and more data, these diagrams became more and more complex. So the story is actually apocryphal, but it makes a point that when you have the wrong paradigm, you're going to keep gathering more data. And when you gather more data, what you're going to end up doing is you're going to end up making the paradigm, or you're going to end up making your mental model more and more complex. We see this a lot in certain fields of science, in certain fields of biology, especially where every new paper that comes out results in a more complex model or more complex pathway. People start talking about these things as if the initial statement of the problem starts becoming more and more complex until you really can't tell what the statement, what the paradigm is, or what the concept is. And we saw this with oncogene addiction. Oncogene addiction used to be the idea that, okay, an oncogene drives a cancer. When you shut the oncogene down, the cancer goes away. Then people were like, well, that's not exactly true. Here's another variation of that oncogenic amnesia, oncogenic shock. By the time they were done with it, you could no longer tell what the paradigm stood for in the first place. So this is a very common problem that you have when a paradigm's in trouble. And the way it works is things become more and more vague and more couched in exceptions. So keep these three things in mind. Important problems remain unsolved. Other paradigms offer competing explanations, and the paradigm keeps getting more complex or vague. Or to put it differently, there are paradoxes that appear within the data that then are that become things that people try to solve by adding more complexity. So with that, I'm now going to move hand over to Rachel, and where she'll be talking about how this applies to the concept to immunology as a whole. I'll just say next slide, please, to me when you need to switch.
Okay, thank you. Yeah, so this is a slide showing a diagram of how different cells in the immune system interact with each other using cytokines, which are chemical messengers. And it's a very, very complex system. And so building this diagram is drawing from a ton of different research results. And I should say as background, like I've done work in math biological modeling of these types of systems where you're kind of pulling in from lots of different research papers. And so I understand the appeal, but it gets very complicated. And if you go to the next slide, Arijit zoomed in on just a piece so you can see better that even a single one of these relationships, such as CD4 positive naive T cell to this what is that even, a ROR1 STAT3 cell down there. There are so many possible cytokines acting on it. There's just kind of a huge number of possible interactions, and they're highly redundant and context dependent. And this is a system that keeps, or understandings of it keep getting more and more complicated as more research is done. They keep discovering new subtypes of subtypes for different immune cells, new cytokines, new interactions. Next slide. So I read this essay by Arijit on R&D discovery, and here he's talking about a pathway in cancer, and he uses the term next Frankencells, which is to describe how these diagrams are cobbled together from a vast array of published papers. And I found that to be a really helpful term. And again, I have done research that involved these kind of Frankencell style mathematical models. And it made me wonder, though, is this a Franken system of what's going on with how we're thinking about the immune system and all these different immune cell cytokine relationships? Yeah, so the current paradigm is becoming increasingly complex and context dependent. And so kind of the hypothesis we're exploring here is that, you can't study a redundant, stochastic, evolving system with reductionist biology. And maybe this is not the most helpful lens or not the right frame that we've been using to look at this. Another sign of a paradigm in crisis is lots of paradoxes. And this shows up kind of all over the place with cytokines and immunology. And so I've taken a few paper titles here, but there are even more if you search. So paradoxes in which particular cytokines are said to have both pro-inflammatory and anti-inflammatory impacts, in which they're shown to both contribute to the growth of cancer, but also the protection of the immune system and stopping cancer. And this poses a lot of, I think, confusion of kind of how do we make sense of all these paradoxes with these different cytokines. Next slide. And it's important to understand that these paradoxes directly impact treatments. And so, as Arijit said in the opening, that kind of these frames for how we're making sense of science and how we make sense of the world really then do have these very practical impacts. And so this is some examples of how these paradoxes around cytokines have impacted treatment for uh, psoriasis, for multiple sclerosis, for COVID-19, and that it's kind of trying to understand, are these cytokines part of the mechanism contributing to the disease and contributing to inflammation, and is blocking them going to help, or actually, is blocking them going to make the disease worse? Has had a definite impact on kind of getting often surprising and contradictory results when they try to put this into practice with treatments. So now I'm going to go through just a few papers on making sense of T cells. And so there are kind of multiple subtypes of T cells. And in this paper, they're looking at the idea of kind of going from the progression from a naive T cell to an exhausted T cell. And I thought it was interesting, this chart along the left, they have different cell markers. So that's what the CD45RA, CD28. Those are all different markers that can be found on the surface of cells. And then along the top, notice that they've got, some of these are CD4 positive T cells, what are referred to as helper T cells, and some are CD8 killer T cells. And note that with the classical definition, you can see that there's kind of this real confusion amongst markers, that in some cases the same markers are actually referring to two different types of cells. In other cases, you can have different combinations of markers referring to the same definition for the cells. And I actually kind of thought the language here is funny, that this paper actually is proposing what they've called here an arbitrary definition of how to try to have the mapping between the definition and the markers on the cell be more consistent. But even there, that's pointing out that there is this real gap in kind of the function of the cells, because the classical definitions did arrive around function and the markers of how we make sense and classify them. Next slide. Thanks. This is a different paper, the who's who of T cell differentiation. And this here is showing this progression of T cells through different stages. And that, well, I should say they're describing it as a progression, and along the bottom they've shown just 4 of the markers and how those markers are expressed or not expressed at different points in the progression. And they're describing it in a more deterministic way. But the question we're bringing to this is, do immune cells go through these stages, which are relatively complex in terms of how the markers are shutting off and on? Or is this dominance of different subclonal populations at different times? And Arijit will share a paper that I think is relevant to that question later. And much of the literature, or some of the literature on this, acknowledges these limitations of using surface markers to define functionality, noting markers can be upregulated on a T cell in a context other than memory, and they may be dynamically expressed at different time points, that some of these transitions are only transient. And also saying that kind of our simple panels no longer are sufficient to capture the complexity of this population, although the overwhelming response seems to be to add more complexity or try to kind of refine the definitions, as opposed to thinking about a different paradigm. This paper looked at, here they've kind of got this T cell developmental hierarchy you can see on the right, with kind of the branching of the different stages in development. And they've taken the approach, though, of looking at these different CPG sites, which can be used for methylation to turn genes off and on. And this is kind of, they end up, even though they're kind of pointing out this that these definitions are confusing and not capturing the complexity, they go in the definition, though, of let's use kind of all these sites to try to classify new definitions. But this shows the complexity that exists kind of even within a category of T cells. And then this, I like this paper. They gathered 10 experts on regulatory T cells, and then they polled them. You can see about 17 different cell markers, and said, do you consider this cell marker to be necessary for, to classify something as a T cell or not. And in the far right column, you can see the number of participants that said, okay, yes, this marker, is necessary to say that something is a regulatory T cell. And so that's a lot of disagreement, and this is among experts of which markers should be included or not. Next. So this is the title of the paper, and this here, they've also even shown the overlap between the different experts, kind of what percent of T cells are you going to capture based on which definition you use, kind of based on the the combinatorics of which markers you're choosing to say what's essential to being a regulatory T cell. Yeah, so the question we're looking at is, rather than considering this a fixed progression through these different stages, could this be described using evolutionary principles?
Thanks, Rachel. And I think you've made the point fairly clearly that you're looking at, these are the hallmarks of a paradigm in crisis. You have increasing complexity, you have paradoxes that are unresolved. So then let's get to the other piece of that, which is there a competing paradigm that could offer a simpler explanation? And so, as you point out, that could well be evolution, right? So just as a quick refresher for evolution, and I know Ryan hates this diagram because it kind of exactly says what evolution isn't. But one aspect of evolution actually is natural selection. And so natural selection was what Darwin put forth in his original book. And there are three components of natural selection per Darwin, right? And one is you have heritable variation. Two is you have selective pressure, meaning something in the landscape, in the fitness landscape, creates something in the environment creates differential, the potential for differential outcomes and unfavorable mutations being selected against. And then finally, over time, you have differential fitness, meaning some subpopulations thrive and others don't. So if you have these three elements, you have natural selection. Again, what we're going to do now is we're going to talk about this in the context of the human evolution, the context of the human immune system, asking the question, where is evolution playing a role, and what is that role? How does that evolutionary role then impact, or how does evolution impact the study of immunology? So it's a very meta conversation. We're really not talking about the evolutionary role of immune systems, and we're not talking about sort of how immune systems shape evolution. We're actually going to talk about how evolution shapes immune systems, in the sense that what are the characteristics of a system under evolution. And then we're also going to talk about evolution at the level of individuals in the immune response and how that impacts the way in which things play out. So when we talk about this at a population level, it begs the question, right? Who is the predator and who is the prey here when we talk about the immune system being under evolution? So obviously, there's two aspects to it. One is that at a population level, humans are the prey. Pathogens, for example, are the predator in this case, and humans are the prey. And then later on, when we talk about the evolutionary impact at an individual level, the equation flips. And it's actually the human immune system that's the predator and the pathogen that's the prey. Obviously, if the human immune system at an individual level loses the fight with the prey, the individual dies in the extreme case. So how does this all play out, right? So one of the strongest forces for selection in the human genome is, in fact, is infection. And so many of the genes associated with managing infections, such as the immune system genes, adaptive immune system genes, and even down to individual pathogens such as Staph aureus, many of these different genes are positively selected for. They're enriched for within the positively selected bucket. So there's a small number of genes that are positively selected for. There is a larger number of genes that have negative selection as well. But in both of these cases, what you see is that selection pressure is felt pretty strongly on genes associated with the immune response or with response to pathogens in general. Making that a little more concrete, we often hear it said, people will often say, well, humans have stopped evolving because now we have, running water or whatever it is, or we have smartphones, so we've stopped evolving. And then people will speculate that, okay, well, what does this mean for us as a species? It turns out we haven't really stopped evolving. If you look at our genomes, a a lot of this is happening in real time, and it's happening really fast. So, for example, malaria exerts a very strong force for positive selection on the human genome. If you look at where malaria is endemic and where you look at, for example, specific alleles, in this case, the sickle cell anemia allele, what you see is that where malaria is endemic is where sickle cell anemia is selected for. And we kind of know this, but this is something we've all studied at some level. If you have a biology background, you've studied it at some point, so it's not surprising. But take a step back and think about what that means. So up until as recently as the mid to late 20th century, malaria was claiming hundreds of millions of lives over that period. And the reason was because you have a lot of infant mortality from malaria. In fact, in the early 20th century, it was as high as 50% of children born in Africa were dying before the age of 4 due to malaria. So by taking out people who are pre-reproductive age and by exerting such a strong selective pressure, malaria actually, by being so fatal, malaria actually exerted a very strong selection pressure. And so different populations have evolved different evolutionary responses independently to malaria. Here's a whole bunch of different alleles that are present in different human populations across the globe where malaria is endemic. And these are red blood cell erythrocyte variations. There are sometimes different antigens, sometimes different enzymes, and sometimes different surface markers, or even hemoglobin. So the sickle cell anemia allele is only one of many different strategies that humans have evolved to cope with malaria. And so what's interesting about this, if you look at this in a practical sense, is that when you look at, for example, the rate at which a specific allele for malaria resistance penetrates the population, as they did in this paper, what they showed was in the Cape Verde Islands over a period of 500 years, which is blazingly fast, the Cape Verdean population, which was originally indigenous, then intermixed with West African individuals. And during that period, the Duffy antigen receptor for chemokines penetrated that population very, very fast. So the evolutionary impact of an infectious disease on our genomes can be enormous. And that's the point being made here. So as I said, at a population level, parasites are the predator, pathogens are the predator, and we are the prey. And it's not a one and done situation, because if you can think of it this way, if malaria exerts a very strong selective pressure on the Cape Verde Island population, and now the Duffy antigen chemokine receptor goes up in its prevalence, what will happen over time, and we saw this when we looked at when we did the series on COVID, was what happens over time is that the predator adapts as well. So you have this constant arms race between predator and prey. Over time, if you look 10,000 years from now, look at this 20,000 years from now, if we continue on the track of welcoming all infectious diseases, to the table, what would happen is that eventually malaria would evolve defenses around these kinds of host defenses. So it's a constant back and forth. It's a constant adversarial game, right? And so this adversarial game has a name for it in the evolutionary world. It's called the Red Queen's race. And so this comes from Alice in Wonderland, from the statement in Alice in Wonderland, where the Red Queen says it takes all the running you can do to stay in the same place. And so the idea here is that both the host and the pathogen are constantly trying to evolve back and forth around each other. And we actually touched on this concept quite a bit in the COVID series, where we talked about what the Red Queen's race means for us as a species. But now this is a slightly more meta conversation, and we're going to talk about what the Red Queen's race means for the study of immunology. Why does this matter in a practical sense when you think about immunology as a discipline? Well, when you think about the Red Queen's race, it's really a dynamic system that's characterized on short timescales by prey-predator dynamics. So as the prey goes up, the predator population starts rising. At some point, the predators start essentially causing a population collapse. That changes the dynamics for the predator and so on, right? And so it oscillates according to Lotka-Volterra prey-predator dynamics. But one of the other interesting things about a Red Queen's race is that in evolutionary terms, it should drive an increase in heterogeneity because of what's called disruptive selection. So the greater diversity you have in the prey population, the harder it is for a predator, in this case, the pathogen, to come in and wipe out the entire population. And so a lot of this comes down to, and we talked about this again in the COVID series, it comes down to a dynamic situation. You can't represent these things on a pathway diagram. And when you think about the dynamics of the system, you then have a lot of different ways of tackling it. We discussed this both in the COVID situation, and we also discussed this for cancer. If you're really trying to treat cancer, you have to think about the OODA loop, the decision cycle, and the faster you can make decisions, the more effective you are. So we'll talk about, again, what this means for the study of immunology and what this means for the design of therapeutics targeting immunological sort of immunologically associated diseases. But the point being that when you are dealing with adversarial coevolution, you're in the realm of evolution, not in the realm of biochemistry. And so in immunology, this Red Queen's race happens as I mentioned, at a multi-scale level, at a population level, as well as at an individual level. Today, we'll discuss in a little more detail what we mean by this. And we'll talk about both what happens at the population level in terms of functional diversity in the human genome as a result of it being selected for, and then also at the individual level, looking at what that means when a human's immune system responds to pathogens. So at a population level, when we talk about this, we talked about the fact that pathogens exert severe selection pressure on the human genome. And this selection pressure, again, this is not news. Everyone knows this. So if you look at that selection pressure, you also know that it manifests in the HLA genes. Now, the HLA genes are genes that present antigens to T cells, for example, and that presentation of antigen to T cell is what drives a chunk of the immune response. Now, it turns out for class 2 HLA genes, which are responsible for common microbial infections, it turns out that individuals have a tremendous amount of diversity. And if you look on the right over here, that table shows you the degree of diversity in responses for different HLA genes against various different common microbial pathogens. So what this says is that when you're dealing with a variety of different microbial pathogens, the human-to-human variation is quite severe. And we sort of know this. Again, everyone kind of knows that HLA alleles are just different. The same is true for class 1 alleles as well. These are very different in different individuals. And again, the heat map shows the degree of diversity. If you look at a wide range of different alleles, which are shown on the in the rows over here, across a wide range of different viruses, what you see is that there's tremendous variability when it comes to the average degree of efficiency with which individuals respond to different pathogens. And so what it comes down to is that if you have a set of any individual with an arbitrary set of HLA alleles, it's just seeing these antigens very differently from one person to the next. Because essentially the amount to which you can actually see them depends very strongly on what alleles you have. Why does this make sense in a Red Queen's race scenario? It makes sense because it means at a population level, it's very difficult for an individual pathogen to evade all of our immune defenses. Now, you could say, well, why don't we simply have all of the antigens that we need to cover all of this space? And we can switch gears and talk a little bit about what that would mean in terms of the math, right? So the point is we only have about 1 billion B cell clonotypes in a person. So if we switch gears and talk about it from the antibody standpoint, it becomes easier to understand this. There's only 1 billion clonotypes possible because we only have about 5 billion B cells in the body. So when you look at antibodies, for example, with 5 billion B cells in the body, you have to sample all of antibody space. And all of antibody space, it turns out, is much, much larger. It's 10 to the power 16 to 10 to the power 18 different configurations. So in this particular paper, which is a really impressive paper, what these guys did was they went in and they sequenced about 3 billion antibodies from 10 different samples. They did deep sequencing, they did annotation, they reassembled them, and they essentially found that, okay, now they have 3 billion antibody heavy chain immune receptor sequences. And so they did a little bit of back of the envelope math, and they said, well, really it should be 1 in a billion antibodies that are overlapping between one person and the next. They actually found a much higher overlap. They found 1%. So the paper title is something like conserved recognition despite extreme diversity. And by conserved recognition, they meant that 1%. But the point is that 99% of any two individuals' antibody repertoires are different from each other. And over here, they looked at 10 individuals. In this paper, they looked at 10 individuals from the same region and roughly the same age. So when they sequence all of the antibodies, each of these circles is a different individual. And you're looking at it's a Venn diagram of overlaps. So the overlaps in these petals of every petal is a different individual. And the overlap in that first sort of first area of overlap is the degree of commonality between these two individuals. And what you can see is these are percentages. So 0.5%, 0.3%, 0.99%. So every human being has a highly individualized adaptive immune repertoire. And evolutionarily speaking, again, this is a good thing because it means you can't wipe out the entire population with one particular pathogen. But it comes from two different things. One is the evolutionary pressure that is placed on us from a Red Queen's race perspective. And it also comes from the fact that you stochastically have to undersample. You have only 1 billion clonotypes, and you have to cover a much, much larger space, which is 10 to the power 18. Now, again, the point here is that only that we would have expected 1 in a billion. We got 1%, which means that the amount of antibody space or the amount of antigen space that actually exists is much smaller. And the thought is that a lot of that is happening because of self-clonal deletion during the maturation of the immune system within individuals. So the vast majority of possible antigen configurations are probably not tolerated and are deleted. So, but nonetheless, it means that any two individuals will never see the same pathogen the same way, neither at the level of the MHCs, nor at the level of the antibodies, and presumably nor at the level of the T cell receptors. So these are all just very different from each other. And not surprisingly, this means, for example, that the effectiveness of vaccine response is very different between individuals. In this particular study, they looked at twin studies, but they looked to see how much of the response or the degree of response for a pair of twins was correlated when it came to things like specific cell frequencies, when it came to serum proteins. And they asked the question both for young twins and old twins. And in doing so, they were able to tease out heritable versus non-heritable factors. And they found effectively that for the most part, the degree of heritability was 50% or less for the vast majority of different pathogens. And so from that, they took away that non-heritable factors dominate the effectiveness of vaccine response. But I will say, if you look at this, you're still seeing a fair amount of heritable factors as well. So the bottom line is there's a huge amount of variability when it comes to individual immune systems. And a chunk of that can be explained by stochasticity, and a chunk of it can be explained by evolution. But either way, it comes down to the same thing, which is no two people are seeing the same antibody, the same antigen the same way, and no two immune systems will respond in the same way, even if everything that happened downstream of there was deterministic. What does this mean in practical terms, though? What it means is that when you're actually looking at individuals and when you're trying to study, for example, the way in which individuals respond to a pathogen, whatever sample you're getting is the sample you're getting. And from one sample to the next, you're not guaranteed that the responses will happen in the same way. And you're not guaranteed that the degree to which the antigen stimulates the immune system is going to be in the same ballpark even. And this is true, by the way, for animal species as well. Humans are not unique in this way. So what that means is that when you talk about reproducibility in studies, this is kind of like a very obvious catch. But taking it a step further, like in a practical sense, what this means is when you're trying to design an intervention, you're trying to design a vaccine, for example, you have to account for inter-individual heterogeneity. And one of the ways you can do that is to understand the heterogeneity as signal and not as noise, because essentially that heterogeneity is baked into the population. So if you want to understand a vaccine effect on populations, you have to have a systematic way of understanding the heterogeneity from one individual to the other and not dismiss it as just noisiness. And we actually did this a number of years ago in a paper that Maddie and I were both on. We took antibody data for COVID for individuals following COVID infection. This was published in the public domain at the time, and we fitted what's called a population PK model to it. And when we fitted the model, what we found was there were fundamentally different conclusions that we were able to draw, where people were looking at the data at the time and saying, well, it's a very long half-life, and as a result, you're going to see very long protection against COVID. We looked at the same data and we found tremendous heterogeneity, inter-individual heterogeneity, and we found that there was a subpopulation that was unprotected from COVID infection in as little as 6 months, after COVID infection. They'd completely lost their protection. So in that paper, which by the way came out in 2021, the preprint came out in 21, I think early 21 or late 2020, we predicted that reinfections, which were rare at the time, were going to end up being a lot more common as a result. And we actually showed in the paper that it was only a matter of not having waited long enough that made reinfections rare. And this was at a time when reinfections being extremely rare was getting talked about a lot. So it goes to show you, when you study population heterogeneity and you take that as being the signal and not the noise, you can draw very fundamentally practically useful conclusions. As we said, for the study of immunology, this really means that your reproducibility question is not so much a reproducibility question anymore, but it's a question of how you're representing the system and how you're looking at the system. So if you study it trying to fit everything to a pathway diagram, you will be disappointed, because no two experimental subjects are seeing the same antigen the same way. At the same time, when you think about this in the design of interventions, looking at downstream markers rather than upstream markers becomes key. Using a kinetic lens becomes key. And using a population modeling approach also is helpful. So now let's take a look at what happens after an antigen is actually engaged with by the immune system. And let's look at what happens within an individual body. In the previous section, we talked about how at a population level, there's tremendous heterogeneity, and that tremendous heterogeneity means even before the immune system is engaged, individuals see antigens in a different way. Now, that assumes that everything that's happening downstream of there is very often when we look at diagrams of what happens when the immune system engages, we see these pathway diagrams that are very neatly drawn out. So the assumption is everything that happens downstream is happening in a deterministic way. But again, we know that there's evolution going on, and we know that the natural selection portion of that has these different components: heritable variation, selective pressure, and differential fitness. So now let's talk about how that plays out when it comes to what the immune system does when it sees a particular pathogen, or how that could be playing out. In simple terms, when you have the immune system engaging with a pathogen, what happens is you get proliferation immediately following the engagement of the pathogen in whichever cell type you're looking at. And that proliferation happens in ways that are both stochastic and heritable. And we'll talk about that now, drilling into a single cell type, because I think it becomes much clearer when you focus on one cell type. So we'll pick memory T cells, and we'll ask the question, what does evolution teach us about memory T cells that the traditional view doesn't? So as Rachel alluded to, T cell nomenclature is a mess. And this is like they got together every single like notable T cell expert in the world. And it's notable that many of the T cell experts that you see on Twitter are not present on this particular consensus statement. But with every single sort of well-known T cell person in the room, they sat down and they said, okay, well, let's make a list of all the things that we think determine whether a T cell is which subpopulation it is. And similar to the exercise that Rachel shared where they had the experts kind of look at this, obviously there's a huge amount of disagreement. And so they came up with this system where they said, okay, well, let's break this out into a set of subscripts. The first subscript is the lineage. The second subscript is the function. And then they had a bunch of other functional subscripts for what the T cells are actually doing. So right off the bat, there's a problem with this, because now you have the traditional nomenclature such as CD4 or CD8, and you have all the stuff that the cell is doing that could be quite different from the traditional expectation, which tells you that this first subscript isn't contributing much to the classification system, because it can exist in any combination. So to make matters worse, what this particular paper proposed was that you can describe your T cell subpopulation in any which way you like. You can say it's a CD4 plus T cell, or you can say it's a CD4 plus TH2DRMO cell, which is, doesn't exactly roll off the tongue. But the point is that these don't necessarily track with each other. And when you read a paper, you're not going to know, if you have a higher level of lineage, you're not going to know what the sublineage is doing. So this is an epistemic landmine. Because you're just not going to know when you go from one paper to the other, what you're actually looking at. So now let's look at a subset of cells. Let's look at memory T cells, for example, right? And let's ask the question, when you look at memory T cells, how does this all play out in practical terms? And is there a simpler way of thinking about memory T cells? So the way people talk about memory T cells is that they're this separate subpopulation and they, essentially start reproducing. And the traditional view is that it was from a different set of precursors that then ended up leading to the memory population being established. Now, the memory population as such is the idea is it's something that has undergone activation when it saw the antigen. It has then differentiated, and then it ends up being long-lived and relatively quiescent and phenotypically diverse. So that's the thought process. But as soon as you start getting into the quote unquote functional markers, all hell breaks loose because very often people start talking about different markers and sort of there's debate around what constitutes memory T cell marker and what doesn't. So a lot of people will talk about CD11, CD95, CD58, CD49D. What's cool about these papers is whenever you read them, they'll be very definitive about a set of markers. You go read a different paper, they'll also be very definitive about the set of markers. When you compare them side by side, they won't be the same set of markers. That's your classic sort of paradigm in crisis warning. So one of the markers that often shows up is CD62L. And CD62L or L-selectin is known for a role in homing to lymph nodes. It actually helps lymphocytes, once activated, head off to lymph nodes. It turns out it's also a critical marker for identifying central memory T cells. And it's thought that central memory T cells, TCM cells, are generally CD62L positive. That often comes with CD44. So all this sounds very neat, right? This is like a textbook type diagram. It looks very clean. No problem here, right? Of course, that's not quite the whole story. So this is a paper from about 15 years ago. 15 years ago that contradicts the figure that you just saw in and the statements that I just made, which are still broadly held today. And this is Peter Doherty, who's a Nobel Prize winner for T cells. And so when you read the language in this paper, just in the way that he's talking about, right? What are memory T cells? How do they develop? A widely accepted idea is that the memory set is derived from the affected population. Current fashion has, if you just read those sort of little highlighted things, imagine you're reading a paper about the mating habits of rabbits, and someone opens it in the introduction with, well, what exactly is a rabbit? Let's talk about what is and isn't a rabbit. You have a problem here. And so this particular paper showed that it's the location and not CD62L expression that was likely to be critical for the memory T's marking up the memory T cell population. There's other papers that have shown similar things. So people say there's a fair amount of evidence, even though CD62L is often associated with memory T cells, that it's not a deterministic marker. So what, in this particular paper, I'm just cutting, pasting a short section from the discussion. What people have shown is that central memory T cell lineage in CD8 cells was unaltered by the entry into lymph nodes as a result of CD62L expression. So when they expressed CD62L, they didn't necessarily increase the amount of central memory T cell commitment. And so therefore they determined that CD62L was not a deterministic marker of T cell differentiation. So how can all these things be true at the same time, right? How does this work exactly? Now, again, as we've talked about, the whole idea is that when you have cells that are reproducing and they are surviving differentially, you should be able to describe that process using evolution. So looking at this weird little conundrum about CD62L got me thinking. And I was like, well, what does the live cell video microscopy have to say about it? Because often that's a great place to start. If you observe what's happening and you have markers, that can teach you a lot of what may be happening under the hood. So lucky for us, there is a paper on exactly that question, and it's a really good paper. So what these guys did was they took blood from mice, they sorted for single naive CD8 T cells, and then they watched those CD8 T cells on coated plates reproducing over time. So they coated the plates with a ligand. And what they did was they watched the cell reproduce over time. Then they tracked all these using image recognition. They tracked the centroids and they could automatically constructed division trees, family trees. So this is a little bit similar to some of the work we talked about, I think, when we were discussing the concept of COVID and cancer. Live cell video microscopy is incredibly powerful in understanding what's happening under the hood because you're actually seeing what the lineages are doing and how it actually plays out in terms of the functional outcomes. So what they did was the first question they asked was, okay, let's look at those cell division times. Let's look at the interdivision time. Let's look at the time, the relationship between interdivision time for daughter and mother or sibling to sibling. And what they found, interestingly enough, was that interdivision sibling times are pretty strongly correlated. So that suggests that there is a heritable component to cell division time. They also looked at daughter-mother interdivision times, and they found those were also quite well correlated. So again, if you think of natural selection as being heritable fitness, the division time between one cycle to the next is that heritable fitness, because what they also showed in this paper was that the overall contribution of cell death in this scenario was very low. So the fitness differences are being driven by differences in inter-cell division time. So when we talk about interdivision time, we're really talking about fitness. And so now what they ended up doing, which was very cool, was they had 3 different theories for how the interdivision time was playing out. What they said was, okay, well, suppose we thought of the model being all of the cells had the same division time, or we thought of a second model where we said all the cells started out dividing fast after being stimulated, and then eventually altered their division time. Or what if we had an initial division time that bifurcated into some cells that divided fast and some that divided slow? And then what they did was they essentially took these videos, they constructed the trees from them, and they compared them to simulated trees generated from each of these different model topologies. So for example, if you have a model topology where some cells are dividing fast and some cells are dividing slow, you can compare the observed to the simulated and say how strong is the evidence using a Bayesian approach. So they did that, and this kind of by comparing their simulated outcomes with their observed outcomes, they were able to essentially come up with an understanding of how strong is the evidence for any one of these 3 models. And what they actually found, interesting enough, was that they needed a 4th model. And the 4th model was the one that explained the data the best. But in the bigger picture, think of it as this approach, where you're simulating on these different kinds of models, and you're looking at the observed data to see how well the observed data lines up with it. And they basically, as I said, added a 4th model, which is that the slow dividing cells had a wide range of division times. And when they did this, what they found was that model number 4, which you see over here in terms of the model evidence, model number 4 had the strongest evidence supporting it. What they found was in model 4, you had an early activated cell population with a certain division time, about 10 hours. Then that split into fast and slow dividing cell populations. And the slow dividing cell populations, if you look over here in panel N, had a high degree of heterogeneity in their division times. So by taking the observed data, which is in gray over here, they were able to, using this kind of an approach, decompose it into a fast dividing and a slow dividing population, as well as a population that was a set of division times that were associated with the initial divisions immediately following simulation. And what they found was, in the way that they designed this, you can see that this is happening between mother and daughter. So after the first few divisions of fast division, or relatively fast division, eventually the division times coalesced into stable division times that were either fast or slow within sublineages descended from the same original parent. And when they did this, they found that this describes the data quite strongly. And they found that when they look at the percentage of trees with significantly distinct branches using this kind of a model, they get kind of this curve that describes the mean data very well. So what they're saying is basically cells start out dividing. After a few divisions, they all start out dividing at the same rate. After a few divisions, some of the progeny start dividing fast and some of the progeny start dividing slow. And that's the model that they propose. And that's what you have over here. Now, that slow dividing population with the very heterogeneous division times is the memory cell population. So what could the mechanism for such a differentiation be? So what they found when they used IL-12 simulation was they found that IL-12 simulation induced CD62L expression. And what they found was those cells that express CD62L more, long story short, panel G, those cells that express CD62L more were also the slow dividing cells. So when they went back and they did FACS and they sorted out the populations for CD62L high and low, and they looked at the population that was CD62L expressing, they found that they had a subpopulation of slow dividing cells. So that's fascinating. What it's saying is that over time over cell divisions, CD62L accumulates in certain cells, and those cells are slowed in the division. And that's happening in response to IL-12 simulation. So what this is saying is that IL-12 simulation is acting as to alter the fitness landscape. CD62 expression on a certain subpopulation changes the fitness of that subpopulation. So it is evolution, but it's evolution happening in response to signals that are being provided in probably a deterministic way between the cytokines and the surface proteins. But the important thing to notice here is that the surface proteins in that case are integrating the signals they're receiving from the cytokines. They're not the functional markers. The functional markers are essentially the functional outcomes are based on the dynamics of the system, meaning certain populations are increasing and certain populations are not increasing as fast. That's the functional outcome. The surface markers are being integrated by the cell. Now, it turns out there are other papers as well that show other functional markers like this that are expressed on memory T cells, and live cell video microscopy with those, which we don't have time to go over today, you can really see with other markers as well. You can see that as the cell accumulates more and more of that surface marker, it slows down in division. So this is not just the one marker, it's multiple. So it's stochastic, it's redundant, it's evolving. And yet it creates a reproducible functional outcome, which is one, there's a subpopulation of cells that accumulates these markers and slows down. And it turns out that reviews that have studied this process for T cells with the live cell video microscopy have shown that there's an actual mechanism by which this is happening, and that is asymmetric cell division. So they've shown that where the immunologic synapse forms, which is the link between the antigen presenting cell and the T cell, where the immunological synapse forms is where certain markers co-localize, and as they co-localize, they change the fate of the cells that divide with the immunological synapse present versus the cell that divides without the synapse present. And the daughter cells that divide without the synapse present become memory cells. The ones that divide with the synapse present divide faster and with less heterogeneity, and they are the effectors. So there's fascinating implications to this. There's layers and layers and layers to this, which we'll get into at another time. But the point is, here's a direct mechanism that explains the stochasticity of CD62L, explains why it's necessary, and explains why all of this is happening in a way that is difficult to represent on a pathway diagram, why you're not going to be able to put all this down in terms of ever being able to come up with a schema for these markers and assign functionality. So basically, what we saw in the paper also, what in that previous paper, was that you have heritable variation, you have selective pressure, and you have differential fitness, all of the key elements of natural selection. So how do you show all this on a biochemical pathway diagram, right? That becomes the question. And so when you have a pathway diagram like this, what you're essentially saying is you're saying that this is a deterministic, pathway-driven, and predictable system, right? APC, antigen presenting cell, meets the T cell, and when it does so, it triggers a bunch of reactions at the cell surface that then lead to signaling downstream. So if this is what is happening, then this is a circuit diagram. And you can study this circuit diagram with biochemistry, you can study it with genetics, you can study it with systems biology, you can study it with AI. And Rachel, this is the topic that's worth coming back to in a little bit. But all of these things are predicated on a deterministic setup. On the other hand, what if T cell behavior was really that T cells look like this, not literally, but that you have one end that receives feeding signals, right, the T cell receptor, and one end that integrates other environmental signals. So the survival signals are coming from the T cell receptor for the most part. Obviously, for memory cells, that's a little bit modified. Whereas for effector cells, the survival signal very much relies on continuous binding to the T cell receptor. And the cell surface markers are providing an altered fitness landscape through the cytokines. So they're a different piece of the animal altogether. I'm showing them at different sides here, but we saw previously that many of the cell surface markers will co-localize with the immunological synapse. But think of this just conceptually, that the animal is doing two different things. It's receiving survival signals for food, which it needs for its continued proliferation, and it's integrating signals from the environment through the cell surface markers. Now, this system is stochastic. The cell is the unit of integration, and it's part of an evolving population. So this is an animal, in effect. And how do you study a population of evolving animals? Well, you look at them. So that's video microscopy. You study their population over time. Population PKPD is one way to do that. But then also there's a whole host of epistemic tools that you can bring to bear on this from disciplines that have been around for 150 years, which you rarely hear about in immunology. Population biology, population genetics, evolution theory. This is just the tip of the iceberg. There's a whole set of conversations to have about how those disciplines will be relevant in this space. But obviously, there's a whole bunch of things you can do when you start looking at formal evolutionary theory and looking at allele frequencies and changes in allele frequencies over time as being not about which specific alleles, but the rate of change of the alleles themselves. You reframe all of the questions and you start thinking about this problem very differently. In very simple terms, what it means is you cannot study these processes using pathway diagrams. You'll be bringing new tools to the table. And of course, there are practical implications for the design of therapeutics as well, which we'll get into. All right, so that's what I have. Over to you.
Wow, very interesting, very interesting. So I think one thing that comes to my mind a lot with these conversations we've had and in these presentations is how there's kind of a recurring theme across different disciplines that are sort of all undergoing this situation sort of around the same time. So you see it with cancer biology, you see it with virology, you see it with neurobiology, where you're studying something that's pretty complex, and there have been successes in a reductionist paradigm to a point, but at some level it starts to introduce those those paradoxes and those discrepancies and anomalies that start to accumulate, and you move into the sort of Kuhnian revolution period. But it's interesting that they're kind of it's all sort of happening at once, and it makes me wonder if you have thoughts on why sometimes those paradigms persist as long as they do, or become as widespread as they are, and as difficult to kind of infiltrate with new thinking as they are, and what the future looks like as far as trying to get people to think differently in those multiple disciplines. So I'm pretty curious about what your thoughts are on that.
It's a big question.
Yeah, it's a big question.
Someone else go.
I know it's a sociological question and a philosophy question and all that.
I mean, I think one factor, and and I'm coming at this as a machine learning practitioner, and I do like machine learning, and I think data science is very useful, but I think that has had a very kind of dominant influence on a lot of fields, often beyond what is maybe useful. And and there is something appealing about being able to gather more and more data, and the idea of like this is information. But so I would say part of it, I think, is the kind of cultural dominance maybe right now of data science, and the the new kind of computational tools that made that possible.
Yeah, no, I agree with that. Maddie?
Yeah, it's reminding me of some of the struggles that we had looking to develop prophylactics and therapeutics for COVID-19 in the grant funding environment at that time. We were approaching the problem taking, again, an evolutionary approach using these different paradigms than what a lot of people were thinking about at that time. And there was a real resistance to funding those concepts. And we were fortunate to be able to self-fund our research and publication at that time. But I think there are institutions, like funding institutions, that are essentially enforcing the existing paradigms and are quite resistant to changes.
And we've seen this, for example, in the Alzheimer's space, where in fact there was actually an Alzheimer's mafia. There was an amyloid mafia that for decades was enforcing sort of the amyloid dogma. And there's plenty of articles out there that have shown how the amyloid mafia literally held back Alzheimer's for 25 years because everything had to be around the amyloid hypothesis. So I think there's that piece of it. Yes, sometimes there's just entrenched institutional resistance, but there's also another piece to this, which I'll layer it in two pieces, because like you, Rachel, I spent some time in the machine learning world, as you're aware. So for me, I've jumped around sort of nomadically from field to field. I've been a cell biologist, I've been a machine learning person, and I've been a pharmacologist. And I eventually sort of got into machine learning because I thought it's cool because, it allows you to think a little more holistically. not necessarily in terms of linear pathways for this happens, then that must happen. So it's kind of cool in that way. But um, very often what I found was we were squeezing blood from stones when we were doing a lot of that machine learning, especially in cancer, because we were force fitting a stochastic story into a deterministic narrative, because machine learning is still, for the most part, assuming that it doesn't deal well with kinetics, it doesn't deal well with the raw stochasticity of the situation. So I will say that at least at some level, some of this, I agree with you, Rachel, is really so the dominance of AI, the dominance of what people are talking about as sort of data science. Um, with data science being used as a catch-all term, because of course you can do data science with evolutionary theory, right? That's also data science. But when people use it, they use it to mean large models or
Yeah,
Mhm.
I was
independent
actually,
models.
I hate the term big data, so I was trying to avoid using it, but
Yeah, yeah,
really it is apt here because
yeah.
really what
Big
is
data,
appealing to
Yeah.
people is quote big data, but it's
100%. And I
yeah.
think I think there's often a belief that okay, if my big data didn't work, then maybe what I need is bigger data.
Mhm.
And we've been spiraling down that. We've been circling that drain for 25 years.
Yeah.
Um, as an early practitioner of machine learning with cancer, I always find it hilarious that people just keep talking about bigger and bigger data sets. If it's an evolutionary process, no amount of data will help you. And we've talked about this from a paradigm standpoint, is when the dominant paradigm was that you could use the stars to tell you useful things about what would happen in your life. Astrologers would map more and more and more stars. The idea being, if you mapped enough stars, you could figure out who should marry whom or when this person's going to die. It turns out that information is not contained in the stars, as Shakespeare pointed out. It turns out that there are things that you can learn from the stars. They're much more mechanistic. We stopped measuring the positions of stars and we started talking about the processes of birth and death. And that's where we went from astrology to astronomy. And I think a lot of the life sciences, unfortunately, are still trapped in that astrology to astronomy transition because they're so wedded to the reductionist approach and the idea that gathering more data will tell you what's going to happen next.
I think there's an interesting aspect of how people perceive the progression or what they perceive as a progression in science as things become more complex. And I think so there's different ways you can look at that. So for folks like I think us, just gauging based on our conversations, when we see, an increasing number of hallmarks or all these sort of asterisks or
Right.
subcategories, and we I think our sense is like there's something wrong fundamentally with the sort of system being used to do this classification or characterization of things. But that's not how that's seen within those disciplines often. I think they look at it as we're developing a more complicated, more nuanced,
More sophisticated.
more sophisticated, framework. So I think back to, the sort of literal, apocryphal as it may be, but the literal epicycles, right, in describing planetary motions and so on, and being increasingly impressed with one's model as it got more and more, epicycles on it.
Right.
Um, and that seems to be what happens in a lot of this sort of
Yeah.
situation, right? Like with the hallmarks of cancer story is a very good example of that, I think, where you start out with, the original intent was here's a bunch of things that are true of cancer and can be used to identify and hopefully treat cancer. And then as these sort of don't work, here's some more. But the notion of hallmark becomes less and less about things that are exclusively and necessarily found in cancer cells to, the most recent iteration had things like phenotypic plasticity, or epigenetic regulation. And those are not traits of cancer. Those are traits of every cell in every multicellular organism. So it's become,
Yeah.
to me, it's like the sort of power of those claims becomes less and less and less as it gets more and more complex and vague. But it's
Yeah.
not
A
perceived like
hallmark, a hallmark of a failing paradigm is that it's unfalsifiable.
it gets to that point, and I think that is perceived from within as sort of a strength, right? Like it's becoming so solid and so comprehensive that
Right.
you can't really it's obviously working well. But in these disciplines, one of the differences in some ways is there's sort of proof in the pudding, test available. Is it leading to productive treatments? And I recently went through a sort of similar reminder in genomics around this when I was teaching a One Health course. And I mentioned, here's what was said about the human genome sequence when it was first, the draft sequence became available at the turn of the century. It's going to tell us everything we need to know about being human. It's going to lead the cure of all, basically the cure to all diseases. None of that happened. And so what we actually need the transcriptome. It's not just the sequence. We actually need the transcriptome,
Right.
and then we'll be able to do that. And then we actually need all the non-coding stuff and the epigenome, and then we'll be able to do all that sort of thing. As opposed to just like, that's just not how it works, right?
That's just
But
not how it
it
works. Yeah,
repeats over and
exactly.
over again every decade or two.
So I think also, I think one of the things is, if you look back at the early model organisms, they were very tractable to reductionist biology. So
Yeah,
there's beautiful work done with lambda phage, for example, around
yeah,
mapping its genetics to what the phage was actually doing. And you could literally map out the decision trees, and you can write lambda phage down to some extent, as a decision tree or as a circuit diagram. And so people coded up lambda, and it's very, very elegant. So I remember studying that stuff when I was in grad school and thinking, wow, this is fascinating to be able to map an organism's behavior to one gene, one function. But
yeah.
in fact, eukaryotes are designed to be robust. Eukaryotic cells don't track that one gene, one function paradigm at all. So the whole thing falls apart, because now you're trying to figure out, okay, well, if you break this one thing, what else happens?
And I think that's what I sort of meant by there's a curse of success in reductionistic biology. It is very successful within its domain,
Yeah,
but often it becomes it shapes the paradigm. So you mentioned sickle cell anemia, excellent example, right? That's one base pair change in the hemoglobin gene, and very clear. You can map out allele frequencies. You can map out all of this, all of the process in a really specific way, because it's the underlying genetics of it are so simple. But the
yeah,
vast majority of diseases are not like that.
yeah.
But the textbook model, and that's the one you learn about first, and right, and repeat that over and over again. Drosophila melanogaster genetics with three chromosomes, and it's like that over and over again, and it really makes it difficult. Those successes, I think, early make it very difficult to not assume that if we just did more of that, like, okay, so it's not one base pair in most genes or most diseases. We need a genome-wide association study with SNPs, but then we'll get it, right? Then we'll get it. It's 10 genes, it's 100 genes. As opposed to, again, the sort of model is wrong. The mental model is wrong. It's really hard to get around that when there's been these like really tantalizing successes early on, but those were successful kind of because they were very well chosen examples.
Yeah, and
I
even the
mean, I
Wilson
think
clock
it's
is right twice a day, right? So it's the Leyden
Well,
jar,
sure.
which is
Yeah, yeah,
the
right,
Leyden jar helps you up to a point, but you can't build a computer with it.
right. Go ahead,
I was
Rachel.
just gonna say, I mean, I think institutions develop a lot of inertia, and part of that is from early successes, but then they've kind of got the methods and way of thinking that, when people spend decades kind of publishing on those, then I mean, it's very hard to do something that's going to refute your earlier work
Right.
or get outside that paradigm. And this complexity is unfortunate in that it kind of just raises the barrier of the institutional moat even higher. Of kind of, it's even harder for outsiders to contribute to this field because there's even more complexity that they don't understand, and so
Yeah,
their viewpoints are discounted. And and with kind of machine learning and data, you even see that as also just kind of the moats around, oh, well, that person doesn't even have access to enough data, or they can't afford enough compute. And so those things
right.
kind of become these further barriers to why kind of outside work can be harder to consider.
And so you raise a great point, which is the moats, right? And so I've seen this a lot in cancer, where a lot of people, when I was a discovery project lead at Takeda, I worked with a lot of people who had all of the signaling pathways at their fingertips, and they could rattle off for you sort of all the crosstalk pathways and all of this, right? And so it created this aura of expertise around them because they knew all the pathways, right? And you see this all the time with immunologists as well. They'll be like, they're very deeply invested in the minutiae of the cell surface markers, even though we all know that the cell surface markers are not a reliable predictor of anything, more or less. But that creates a mode. It creates sort of a priesthood of people who are qualified to talk about this because they know all the cell surface markers. And if you take a step back and say, well, gee, the cell surface markers are probably the wrong end of the animal, because if you view it from an evolutionary standpoint, you'd be much more interested in asking the question, what is the differential survival? What is the differential fitness of the T cell receptors? Because that's going to teach you a lot more. That's I think it becomes a very difficult proposition that all of a sudden all of the data that has been gathered may not actually be dealing with the problem at hand. And it's true I find that in cancer as well. When we talk to people, they nod their head and they're like, yeah, that sounds right. I mean, it is evolution. That makes sense. And then again, they'll go back to doing their whatever they were thinking with their signaling molecules.
Trish Greenhall, a professor in the UK, has talked about with considering airborne spread that the group that was kind of most resistant to the idea was infectious disease specialist, and that feels backwards initially because it's like, oh, but they focus on infectious diseases, but it's also like, oh, okay, well they spent decades being kind of like brought up in this paradigm that wasn't focused on airborne transmission, and so it is or can be, and she's published on this, like can be hardest for them to accept this new way of thinking, and they kind of have the most, I guess, built up traditional paradigm that they're using that doesn't account for that.
Yeah, I think we saw that as well. And again, I try not to be to give undue weight to the most vocal folks, but they are the ones that tend to be interviewed and whatever. But epidemiologists and virologists got it the most wrong, in many ways around evolution of the virus, right? Of SARS-CoV-2 and just in general. And I still see stuff like, we keep being surprised by these, major jumps in the evolution, and I sort of go, really? I mean, that's exactly what you'd expect, and that's been the pattern for several years. But you feel like, and I didn't know this going into sort of the experience of thinking about viral evolution during a pandemic, but it's very typological. It's very coronaviruses do this and this one does this, and it's really not. It's sort of almost pre-evolutionary, right? Like it's sort of like taxonomy was before evolution.
You see that exactly repeated with the T cell population.
That's exactly right.
That's exactly what they're doing. They spend the whole day doing library science with the markers, going, well, does this make it a CTM cell or not? What about that marker? And it's like, okay, that's one way to think about the problem. But if you reframe your thinking, you would be approaching the problem with a bunch of very different analytical tools.
I mean, there's a lot of this is repeated. I mean, I don't want to make it sound like evolutionary biology or biodiversity science or what have you have not gone through all of these things too, because it absolutely did. It just was quite a while ago. But you had the whole Linnaean classification system pre-evolutionary, right? And so you have lots of it works, right? It's your Leyden jar sort of thing. A lot of it works, but a lot of the decisions also end up being kind of arbitrary, and that you have no real independent manner by which to weight different characters that you're using, right? So initially Linnaeus initially had whales in with fish because it's like, okay, they live in the water, they swim, they have this body shape. Why wouldn't you choose that? On what basis would you choose milk production as being more of a weightier character? Well, history is sort of the decider now, right? So in other words, you want phylogenetics, you want evolutionary relationships, and you specifically are trying not to be fooled by convergent evolution, the similar traits that are, so there's historical component. So that the live video microscopy approach that allows you to have some of that seems like it would be a productive way, rather than just looking at all the characteristics and trying to find bins that fit everything, because it's not going to happen in a case where things can happen via different pathways. Try to come up with almost a phylogenetic classification, that looks at historical branching to put things into categories. But that's where we've been. Like we've been that way with trying to classify living things, and we still debate about what species are or aren't. You have to decide on operational definitions. But it feels like when people talk about coronaviruses do this or whatever, it's lacking this notion of they can change, and you haven't seen everything, right? You've seen a few. And then when you're trying to understand why things become diverse in the way that they have, if you're starting with every character as equally relevant, right? As sort of Linnaean, you're just trying to make your best decisions about where to put things, which bins to put things in, and you're going to have to make arbitrary decisions about which characters to weight more heavily. The difference now is we have some independent information, either genomic comparisons or ultimately shared history. And I don't know if that's happened in sort of the what I see when you talk about the implications of the live cell microscopy and the sort of approach of tracking those bifurcations, is that it gives you, now you can start to talk about, like, maybe what there are branching options, not equally weighted characters all at the endpoints. And that, so what's useful is it's not just that there's a framework that can provide a different set of explanations. It may give you a different foundation for how you approach some of those questions about what you're even trying to describe, right?
Yeah, so that's a great point. And so I wanted to actually pass the question back to the group, which is, okay, it's all great to say a different way of thinking about it, but what are the practical implications of this? Maddie, you want to jump in?
Yeah, well, we've been chewing on practical implications for therapeutics in the immunology space. And I think the maybe simplest one to think about is autoimmune disease. I think that this would then suggest that autoimmune disease is a failure of the evolutionary process, a failure to prune nodes that shouldn't be there. And so that, I think, reframes the treatment paradigm in terms of how do you get evolution back on track? How do you go back to pruning
Within the
those
individual.
within the individual? Yeah. Yeah.
Not
Not at the population level.
With direct parallels to cancer as an evolutionary.
Yeah.
Right.
Yeah. It makes autoimmunity look much more like cancer. And I think some people have commented on this. It also means that autoimmune diseases are not many different diseases. They may be one disease with the same underlying etiology, but manifesting differently depending on which T cell receptors are the ones that are driving aberrant proliferation.
I think another one that you raised that maybe you can expand on a bit is vaccine design, or vaccine design or implementation, I suppose. So what comes to mind is the current flu situation. And an article which is less about super flu. It's more about, look, there's variability in flu and it evolves, and those are sort of core things to understand, as opposed to flu does this. But I read a discussion around that, and one of the quotes from the expert interviewed was, it's really surprising that you see these really, these very different impacts across relatively close regions. In one place, in one country, you're getting this massive wave, and in another country, it's not doing that. Well, I mean, again, that sort of feels like you're assuming all humans are the same and will respond the same, and right, it's just missing that core piece that you mentioned. But if you're trying to counteract that, what do you see as sort of implications, practical implications for that in terms of vaccine design or distribution or tracking? Do we measure the variability among people in some way? How do we get on top of that? Like that one study was amazing, but it was 10 people.
At a high level, Maddie?
Maddie,
yeah.
It's a really good question. It just has many layers.
Yeah. It does. Yeah. So I will speak to at least part of that question. Something I've been chewing on is we think about how, the COVID vaccines, how the flu vaccines are designed right now.
Well, COVID is just whatever the current
whatever, and flu, they're doing some kind
of flu, yeah.
Evolving the virus in a chicken egg or something like that. But more or less, both paradigms are kind of chasing the variation that exists. I think that what Rachel and Arijit have just laid out suggests that we should be focusing more on, rather than, what is going to come out next from the virus, how do you set the immune system up to deal with, variation? So we touched on some of this in some of the papers we wrote on a very high level, thinking about boosting schedules for the COVID vaccines. I think basically what instead you should be focused on is how do I make the antibody repertoire or the T cell repertoire as relevant, more diverse, more adaptable. In the context of the Red Queen's race, kind of thinking about, I don't know what's coming next, but I know that more diversity, more T cells, more antibodies in general will sort of tip the balance toward the immune system beating out whatever the virus does next.
Yeah. So that's a great answer, Manny. And I think one other thing I would add to that is that when you really think about how to apply evolutionary theory to these things, there's layers and layers to it. And I'll give you some simple examples. Even if you're thinking about how often to do an update, you can really apply Red Queen's theory to that question to get better answers. So again, this is early days. All we're saying is, look, we should be studying this using evolutionary theory. What we learn, where that journey takes us is unknown right now. But right now, this is just almost a plea, not just to the scientific community, but also to the public to say, hey, if you think about this as evolution, it may give you some different answers. And hopefully, this kind of a attack, if we can evangelize it, hopefully people pick up on it. It's a whole new way of thinking about the field. And so hopefully that gives us some new ammunition. But obviously there's a lot more that can be done. I have a question for you, Rachel. Maybe this question and everyone's responses to it. We wind up with this because we're well past the hour. What do you think that this view of immunology says for AI? And what do you think it says, for example, for virtual cell kinds of efforts, the Chan Zuckerberg stuff? Where do you think that's all headed when you look at sort of how we're describing just the immune system alone in the context of like the hopes of building enough data to really be able to predict what's going to happen next?
I mean, I think that data is still very useful. To me, the kind of the highlight is the paradigm that you're putting into, in some ways, really forces or limits what types of conclusions are possible. And so I think that, and this is even, I think often it's kind of glossed over, but in a machine learning model, like you are making choices about kind of, what format the input's going to have, and then what you expect the output to look like, and kind of what, in a way, you are kind of constraining the shape of the problem by decisions that go into how it's structured. And I mean, this is another interest of mine, that a lot of this work around what data is used is often kind of seen as invisible, and people think like, oh, this is just totally objective, you're just doing what the data says. But a number of the choices you make in what data you gather and how you process it, and then the kind of architecture of your model are imposing a worldview in a sense. And so I think that this view can be very compatible with machine learning. I mean, there is a lot of stochasticity and probability that goes into machine learning, but I think that this is going to probably force probably a helpful inspection of like, okay, like let's think specifically about what data we're using and how what data we even want to gather. Because another thing that gets kind of overlooked is people just kind of use the data that they have available, as opposed to thinking like, well, what is my question actually, and what would be useful for solving the underlying question. So I think that machine learning will still be useful for this, but I think this does kind of point to some necessary reflections on kind of how problems are framed.
I really like that point you said at the end about the kinds of data that are available, and it sort of goes even a step farther, which is the framework will determine what kinds of data collection get done. And I think when you start to even just do what we're doing here, which is just talk about an alternative way of thinking about things, it may reveal the lack of data that allow you to test some of those ideas and allow you to propose what kinds of data acquisition needs to happen in order to test those ideas. And then now suddenly you may have something that's not just more and more and more of the same, right? And I think, Arijit mentioned right at the end, I think, bringing in population genetics and other evolutionary kinds of frameworks. And I think in that case, if you were to sort of start building simulations or models and say, what's the what kind of selection coefficient do you need across different cell lineages to see this outcome, or what determines that adaptive landscape. What is the most important factor in determining that? You start to make these predictions, and then it would let you know what kind of data you need in order to test those predictions. And suddenly you're dealing with a really different set of information, which would be amazing, I think, right in the end. Rather than just we really just need to add more to this circuit diagram or whatever. So yeah, I think that is where it could very well go, and that's an interesting example of the model driving what kinds of data we do, rather than it being technology driven, and then you have to build your models based on whatever we've collected, which is sort of how a lot of the reductionist stuff goes, right? We're just going to sequence more and more genomes, and then you can figure out with your models what matters, right? Not necessarily that a super productive way of doing it.
Agreed. All
Cool.
right.
Well, so I think, yeah, we should probably wrap up. It's super interesting. It feels like there's a lot more to explore on this topic, as with the other stuff that we've been discussing, but a really good introduction. And I think what I really like about these sorts of things is we're not talking about, an infinite list. What we're not doing is just more and more types of T cells, being named. We're saying, look, there's these core concepts, these core mechanisms that are being applied in very different contexts, but the core is very similar across them. And to me, that's a very simplifying kind of approach, right? Conceptually simplifying. And so I think that hopefully people who have watched the previous videos and this one are starting to see those themes reemerge over and over again, rather than just, wow, this is just one thing after another. That's the whole problem we're trying to address, actually, is that it's just these long lists of individual observations we're trying to get away from, and more into a unifying set of concepts. So I assume there will probably be some more discussion on immunology as a topic, and there's various other things that we want to get to as well, but we should probably leave it there for this one. Thanks everybody so much for your insights and for the really interesting presentation. And I hope people will join us when we continue on this topic.
Thanks, everyone.
Cheers.
Thank you.